are going to discuss transcription and translation. So what is
Yes, ma'am.
transcription? Formation of RNA from DNA. Isn't it? The messenger RNA
Yes, ma'am.
from DNA, that is nothing but transcription. Translation is nothing but whatever information comes in the form of mRNA, it is translated and it is helping in the formation of proteins. It's a lengthy chapter. We will see stepwise for your better understanding. Okay.
Yes, ma'am.
So central dogma of life in the previous chapter also it was there. DNA undergoes replication to form daughter DNA. From DNA we see the formation of RNA by the mechanism of transcription, and RNA is forming protein with the mechanism of translation, right? Genome, proteome, all those things you need not worry, and transcription in prokaryotes also we need not worry much. Okay. Yeah. So transcription in eukaryotes, this is very, very important. So it's a complicated process. And now let us see, like we had DNA polymerases for the synthesis of like daughter DNA in the during the process of replication. Similarly, for the synthesis of RNA from DNA, we have certain enzymes which are RNA polymerases. So we have different RNA polymerase like RNA polymerase 1, 2 and 3. So you can see here, see this is DNA strand, one is running in 3 prime 5 prime and the other is running in 5 prime 3 prime direction. Double stranded DNA molecule you are finding. So we have certain enzymes like RNA polymerase. It is identifying a particular area of the DNA. When this enzyme acts, we will see the formation of rRNA, ribosomal RNA. Then RNA polymerase 2. This is helping in the synthesis of messenger RNA. Okay. Then we have RNA polymerase 3, which is actually helping in the synthesis of transverse RNA. So these are the RNA polymerases which are actually helping in the synthesis of different types of RNAs. Now, because the DNA structure is highly complicated. Inside the nucleus, we have chromosomes, which are highly coiled structures, and within these chromosomes also, a particular area should be identified, like which has to undergo transcription. So for which, that means there should be a special identification site. That means a special sequence of the nucleic acids which are identified by these RNA polymerases, so that they will be able to translate, like transcript. That means the formation of a piece of RNA from that particular sequence of DNA. Now, like that, we have actually like Tata box and Hogness box, which are actually called as promoter sites. Okay. So what are these like sequence like Tata and Hogness box? Let us see. See here. See, this is a double stranded DNA you are finding here. One is 5 prime 3 prime, the other is 3 prime 5 prime. Okay. Now if you just see genetic material, anyhow double stranded DNA is all made up of nucleic acids only, A, T, G, C, C, T, G, A, anything, like you have so many like nitrogen bases in both these strands. So wherever you are finding in that like millions of nitrogen bases, if you find the arrangement of the nucleic acid like GG, CC, AA, TC. So this is actually called as cat box, which is generally 70 base pair away from 5 prime direction if you just count. So if the RNA polymerases are finding cat box, so this is the place where they will be transcripted. That means this will be identified and it will be read and it will be synthesized in the form of mRNA. One more like identification thing what we see in this double stranded DNA, apart from cat box, we also have Hogness box, which are 25 base pair away from 3 prime direction, and the order is AT, that is Hogness box. So these are the promoter regions, so which are identified by these RNA polymerases, Tata box and Hogness box. Okay, so which will be helping for the identification of a sequence of the nucleic acids to be read and helps in the synthesis of different types of RNA. So now coming to the initiation of transcription, same thing what I told you, like see molecular events, you have chromatin which is so much highly dense. Okay, so promoter sequence we have, like that is Hogness box and the Tata box. Apart from that, inside the nucleic acid, inside these chromosomes, we have certain enhancers in the sense there are certain substances which will make this cat box and Hogness box more visible. Those are called as like enhancers. So stimulation of transcription by enhancers. So large number of transcription factors interact with eukaryotic promoter regions in humans. Such 6 transcription factors have been identified. Okay, so you can see it is all TF II. Only thing differs is DABFEH. Okay, so these are nothing but enhancers which will allow the Hogness box and Tata box to be visible more clearly. Okay, they bind each other in turn to the enzyme RNA polymerase. Okay, so these are transcription factors. Enhancers will increase gene expression by 100-fold because it's a very much complex area, no, the chromatin. So a particular area has to be identified. So all these factors are helping. Okay, transcription factors, enhancers. Now, once the mRNA or tRNA or RNA or RNA is formed, it will have so many things which are actually not required in its structure. So it undergoes certain changes which are actually called as post-transcriptional modification. So in this post-transcriptional modification, the heterogeneous RNA is nothing but crude RNA, as though it is jaggery, which is now being refined as sugar, something like that. So it has all chemical bondings which are not required. There are certain chemical like elements also which are removed. So the crude form of RNA which has been identified and synthesized by RNA polymerases has to be refined. So, and that crude form is heterogeneous RNA. So, when it comes from the nucleus into the cytoplasm, so this crude RNA will undergo certain modifications at head end and also tail end. So, this is called as end modification. Okay. So, 5 capping. So, 5 end of mRNA is capped with 7 methyl guanosine. Actually, this methyl group is again coming from always one carbon donor. What is that? We have SAM, isn't it? Acetylinosyl methionine will be transferring this methyl group to 7-methyl guanosine, which is serving as a cap, as one of the modifications after the formation of heterogeneous RNA. Poly A tail is nothing but a structure which is actually helping, tail in the sense as though sperm has tail, no, which helps in motility. Similarly, large number of eukaryotic mRNAs, they produce adenine nucleotide chain at 3 end, so which is actually seen during transcription. In later the poly A chain, it is just actually getting disappearing because once it has moved into cytoplasm, its purpose is over. Okay, next, the heterogeneous RNA, if you observe this structure, so it has like an area which is coding for amino acid, an area which is not at all coding for amino acid. It is something like a dummy, nothing is there. So like in case of like introns and their removal, introns are the places which are not coding for any amino acids, so the introns are removed and the exons are actually patched up. So with the removal of introns is carried by this NERVs, SNRNPS NERVs, where ATP is utilized and it is converted to ADP and high energy phosphate. Okay, so intron removed, exon, it is just getting binded up. Okay, so that is one change you are finding. So that is introns and their removal. So these are the changes. Then different mRNAs produced by alternate splicing. Mm, see, so as I told you, this picture will give you as a summary of what we have been studying till now. See, this is chromosome. Here we have this centromere. We have P arm and Q arm. We know inside the chromosome we have dense chromatin. Like so much of information is there inside the chromosome. A particular area of the chromosome is being identified with the help of that Harkness box and Tata box. Nothing but. Okay, and it is being read. Okay, 1.5 into 10 to 8, the power of 8 base pair if you just consider it. So these are all gene clusters. Okay, 1.5.
Yes, ma'am.
Now what you are seeing is, so once the reading goes on with the help of these RNA polymerases, you will see heterogeneous RNA is found, right? So this heterogeneous RNA will have introns, exons, useful things, useless things, everything is there in the structure. So if you just count here, we have 8 exons and 7 introns, and we know introns are not useful, they have to be removed, right? Only exons have to be patched up. So the white areas are actually representing the introns, and we need only exons here. So this is primary transcript, which is nothing but heterogeneous mRNA, right? So you are finding the introns are removed stepwise, and whatever RNA we require, which is going to help in protein synthesis, is finally formed. And this is what is called as messenger RNA, mRNA or messenger RNA. Okay, so this is what is happening, like in all these things what we have studied, introns and the removal, different mRNAs produced by alternate splicing. That means whatever splicing you are finding here, so the arrangement of exons will be little different, with the help of which you will see mRNA, tRNA, rRNA will be synthesized. Like we found inhibitors for replication to stop the cell cycle, cell multiplication, to prevent cancer, I said. Similarly,
Yes,
here, if you
ma'am.
don't want the production of like proteins, then we have to go with inhibitors of transcription by making use of certain drugs like antibiotics and toxins. Okay, beta? Oh, antibiotics can be actinomycin, rifampicin. Basically, we use it as antibiotic only to treat tuberculosis
Yes,
and
ma'am.
also leprosy. Okay, alpha amanitin. These are the drugs which are generally used to prevent transcription. Okay, beta? Now we have reverse transcription. Reverse transcription in the sense, basically from DNA you have seen the synthesis of RNA. But from RNA, if DNA is synthesized, then you call it as reverse transcription. Such reverse transcription usually takes place in case of viruses. Okay, and such viruses are called as retroviruses. Reverse. Retro means reverse. So, and they are cancer causing, hence they are called as oncogenic viruses. Okay, so enzyme here, what is the enzyme which is required? Reverse transcriptase. This is the enzyme which is responsible for the formation of DNA from RNA. Okay, so DNA is now being synthesized from RNA. So that is reverse transcription. Now translation is again one more lengthy topic, beta. We will take it as a separate class tomorrow. Is it all right?
Yes, ma'am.
Huh?
Yes, yes, ma'am.
Okay.